Good day, everyone. This is Vijay Yadav. Today, we'll be discussing about the topic propagation technologies of horticulture crops. In the last unit, Introduction to Horticulture, you might have learned what is a horticulture, what are different crops included in it, what is the difference between the cereal crops and horticulture crops. Now, we will see what are the different ways to multiply these crops. So, by the end of the lecture, you will understand propagation in horticulture crops and what are the different methods of propagation in horticulture crops, including micropropagation, and what are the different advantages and disadvantages of different propagation technologies. So, at first, let us understand what plant propagation by definition means. It is actually a purposeful act of reproducing plants. So, this is not a new thing and it has been practiced from a long time ago, maybe similar to the time when this human civilization started, maybe 10,000 years ago. And human being has been observing the nature and has adapted different techniques and also introduced different innovations in that so that he can multiply the plants with a specific value to the mankind. The main aim later was to establish new gardens, new orchards, and new plantlings when the man started settling at certain places. So, unlike the cereal crops, which are mostly propagated by seeds, horticulture crops are propagated by various techniques. We will be seeing the different techniques in the coming slides. And the choice of the propagation technique, what technique has to be used, again depends upon the crop you're choosing, the cultivar of the crop, the climatic conditions where you're going to grow, the soil conditions, and the biotic factors such as the diseases or you have your pest and you have your abiotic factors like temperature, humidity, and many others. So we'll see what are the different propagation methods. So broadly, the propagation methods can be divided into two types. That is the sexual method and another is the asexual method. Coming to the sexual method, as the term says, over here the propagation is done by the product of the sexual reproduction. That is the seed, which is formed by the fusion of male and female gametes. So by the fusion of male and female gametes, by the process of fertilization, you have an end product that is embryo. And from the embryo, you have something called as a radicle, which grows into the root system. And then you have hypocotyle or plumule, which grows into the shoot system, which can be seen in the picture over here. And then talking about the asexual method. So over here, the propagation is done by any vegetative part, just other than seed. So that's the reason it's called as asexual method. So it's also called as a vegetative method because over here no sexual reproduction is involved. It is also called as a clonal propagation because the products of this propagation method are similar to the parent plant because there were no recombinations happening. So all the plants which are produced have a similar qualities, which is ideal condition for commercial cultivation of fruits or vegetables or any horticulture crop. In the picture, you can see the peach scion, and in the red circle, you see the graft union, which is tied with a grafting tape. And the peach scion was grafted on the wild plum rootstock. This grafting is also one of the method of asexual propagation. So coming to the advantages and disadvantages of the sexual method first. So the most important advantage is that it is easy to propagate using sexual method. Just like for example, you are having a fruit and there are some seeds which you got from the fruit. You can just wash them off and then put it in the soil and they start growing. It's as easy as that. And another thing is if they're stored properly in a proper conditions, they can be stored for a very long period of time. So there was a case where date palm seeds were found during excavations in a fortress near the Red Sea in Israel. And then when they planted those date palm seed, surprisingly, it germinated. So you can understand how long a seed can be stored if they are stored in a proper storage conditions. And then another thing is the long life of the plant. So when you're comparing with the asexually propagated plants, so these have a relatively a long life, and this is because of the extended root system, the hardy nature of the plant, and also the vigorous growth. And another thing is this is the only method by which you can produce rootstocks. So another thing is, and this is because they have a very strong root system, which comes with either extended root system, and this is again not found in asexually propagated. Like if you are having a plant through the cuttings, you don't have a very strong root system. They are the adventitious root system, and they are not very strong. And the seed propagated ones are relatively free from viruses, and it's relatively safe to grow with the seed if you are having any issues with the virus. The main disadvantage is the non-uniformity. Like because of the sexual reproduction, there's a lot of recombination happening, and the different characters are produced. So the two plants are not similar, which is not an ideal condition for the commercial cultivation. And they will have a relatively long juvenile period. Like for example, if you are growing a mango by a seed, it might take 7-8 years to give you a commercial yield. While if it is an asexual propagation, maybe it will start from 1 or 2 years age. Another thing is it grows very vigorous. So when it grows vigorous, so there will be a need for regular pruning and training, which can be a tedious job. So another issue with the sexually propagated method is the seed viability. So seed viability is the time period for which the seed can germinate. Like for example, in some of the crops, it is so short, like mango, citrus, and lychee. It could be the seed viability period could be for the days. So you have to plant within those period. Only then you will have a germination. You will have a plant ready, or else it's of no use. It won't germinate. And another issue with the seed propagation is the seed dormancy. So there are different conditions why the seed dormancy happens, and we need to provide a different seed treatments to overcome the seed dormancy and encourage the germ. And then in the pictures you can see the jujube seed with a very long the tap root, the root which is coming out of during the germination. This will develop into a taproot system and an extensive root system. In the second picture, you can see the smoking treatment, which is sort of necessary for certain Australian native crops. Only then these seeds will start germinating. So now coming back to the asexual method. Asexual method again can be of different types. It can be grafting, budding, layering, or cuttings, depending upon how you do that and depending upon the necessity or what is your requirement. So coming to the advantages, as I mentioned in the sexual one, they asexual propagation is produces the plants which are relatively uniform and they are similar to the parent plant because there is no recombination or what is a fusion of gametes are happening. So here the plants produced are all uniform to the parent plant and they have a relatively less juvenile period, like as I mentioned earlier. So the mango plant, if you are producing with the grafting, it will start it will start giving the fruits maybe in a 1 or 2 years age. And then in some of the crops, there is no other way other than asexual propagation, like for example, banana. In case of banana, there are no seeds produced because of a character called as a parthenocarpy, and there is no other way other than asexual propagation to multiply banana. And then another important advantage of asexual propagation, especially grafting and budding, is you get the combined advantages of both rootstock as well as scion. So this gives a chance for adaptability and also while increasing the productivity. So and these are relatively less vigorous. So you maintaining or training and pruning is relatively easy when you're comparing with your sexually propagated one. So the main disadvantages comes out to be the chances for the virus is more in case of asexual method, and there are quite an evidences like there was an incident where banana crop was totally like a gross Michael cultivar of banana crop was totally destroyed because of the Panama wilt disease, and there is no resistant or tolerant varieties there, so it was totally devastated. And similarly, the banana bunch top virus, which has significantly reduced the production of banana in Southeast Asia, South Asia, and Middle East countries. And they have a relatively shorter life in case of perennials, like if a seed propagated tree, if it stays for about 50-60 years, maybe the same crop, if it is propagated by sexual method, might live for 10 or 15 years in being productive. And these methods of grafting, budding, and sometimes layering might require a specific skill to do. Like you can see in this picture, the asexual method can be as simple as a serpentine layering, where you just make a notch on the stem and you put it under the media. Then you put a weight or you peg it into the soil. Or another way it can also be, it may also require some skills, like where you take a scion and you fix it into the rootstock, and then you tie it properly so that the both of the KMBMs or both of them join and start growing together. So this, if it is not done properly, so might fail and the scion might die off. So the even a very good conditions, the success rate, if it's not done properly, you might have a very low success rate. And again, not all scions and stalks are compatible to each other. Like for example, you have a certain rootstock, it may not be compatible with a scion, and it might show the effects of incompatibility early, where the scion stick dies off, or the symptoms can be shown at the later stages, as you can see in the picture. The first picture is of an apple tree, where you can see that swelling thing happening over just before the union, just above the union. And over here, the citrus, you can see the swelling happening over here. This is the symptoms of the graft incompatibility happening at the later stages. Sometimes it can be seriously affecting the productivity of the crop, or in other times it may not be very significant. But yeah, this is the symptoms of a graft incompatibility. Next, coming to the asexual method of micropropagation. So micropropagation, as the term says, it is propagation using a small tissue or a smell, and this is done by exploiting the property of totipotency, which is present in the plant cells. What are the main advantages? Either you can produce rapid multiplication is possible through the micropropagation, and it is free from the viruses. And if there is any tissue which is not germinating or some condition it is not able to produce by the seed or whatsoever, so you can rescue that embryo and produce in a lab condition and multiply that plant or plantling. And as the propagules are small in size, it is easy to transport, and it's relatively easy method of propagation or multiplication. And there are disadvantages too. So it might require a very expensive facilities, and then it needs the technical skills. And if it is not done properly, when it is transferred to the normal condition, the plant might fail. And there's a possibility of mutations, like a sudden genetic changes, which can be undesirable, and a simple contamination can damage the whole lot of plants. So care has to be taken. So these are pictures where the first picture shows the callus development from the explant, that is the small tissue which is taken and grown in the media. Second picture is once the explant is developed, so it is allowed to grow into the different organ, shoot system and root system. And then you can see they are transferred into the protease, and then they will be transferred into the field after the hardening. So in the today's discussion, so we understood that plant propagation is not a new thing. It is originated from thousands of years ago. And the application and recent advances uh, is helping to do it in a better way. The sexual propagation is the most common in the nature, and when you're doing the asexual propagation method, it is mostly suitable for the commercial orchard because of the uniform characteristics. And talking about the crop-specific propagation technique, a person need to have a good knowledge about it if you want to have a commercial cultivation, uh, uh, how to choose and what propagation technique to choose. And then micropropagation technique can be used to rapidly multiply virus-free plantlings and in a very protected or condition. So these are the few suggested readings. So you can just go through this. And if you have any questions, please feel free to mail me over here at this email. And thank you very much.